Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 4 from our Genetics Essentials, Concepts and Connections textbook, 4th edition. This chapter deals with extensions and modifications of basic principles of genetics. What this chapter is essentially going to show us is that not all traits are inherited in a Mendelian fashion with a clear dominant recessive pattern where you would do a monohybrid cross and you would see 3-to-1 phenotypic ratio, a 1-to-2-to-1 genotypic ratio every time. There are exceptions, and these exceptions are called non-Mendelian inherited traits. And there are good reasons for these exceptions. This does not disprove Mendel, but it shows exceptions, and we are going to explore in this chapter what those exceptions are and how they arise. And one of the main ways they arise is by dealing with the sex chromosomes, because the sex chromosomes are different in the different sexes of a species, right? So you've heard of the X and Y chromosome. The X chromosome has to do with female-ness and the Y chromosome with male-ness. Well, the Y chromosome is actually a very stubby, short chromosome, as you can see here on the left in this image 4.1. There's only a handful of useful genes on this chromosome, and the main one being SRY, which directs maleness. Now, the X chromosome is much longer, and it has a host of different genes, so it has a lot more useful genetic information. Now, if there are these genes, if there are problems with these genes on the X chromosome, well, they're going to impact females differently than males. Okay, so we're going to get into that in this chapter. And there's other reasons that you would have non-Mendelian inherited traits as well, but a lot of them have to do with the sex chromosomes. So again, you have sex chromosomes, the X and Y. During meiosis, the X and Y chromosomes can pair. Even though they're not homologous, they can still pair at the telomeric regions. There's these regions where they can still pair. And during sexual reproduction, when you have meiosis to form the gametes, remember the gametes are haploid. Those gametes come together during fertilization to form the diploid zygote, and the life cycle continues this way. Remember that males have X and Y chromosomes. Females of different species have XX chromosomes, right? So it is the male who dictates the sex determination of the offspring, because it's the male that has either an X or a Y to donate to the sperm. The females have 2 X chromosomes, so they cannot direct maleness through a Y chromosome. So it's the sperm, essentially it's the sperm that can dictate a male or a female embryo or zygote. So concept check time. Concept check number 1. What process causes the genetic variation seen in offspring produced by sexual reproduction? Well, we touched on this in a previous chapter, right? We said that it's meiosis that gives you genetic variability, right? Remember that? By way of crossing over and independent assortment, that's where the genetic variation is introduced in populations. It's during meiosis, so meiosis is the answer here. Now, not all eukaryotic creatures use the XY system for determining sex. Some have either 2 X chromosomes for female and an X by itself, they call it standalone X, for maleness. So for example, in grasshoppers you would see 2 X chromosomes directs the sex determination for female, and 1 single X chromosome would direct the grasshopper to be male. Now, in humans, we have the XXXY system. two X chromosomes female and an XY male. And you would see this in most mammals. And again, the Y chromosome is very short and stubby, lacking very many genes at all, whereas the X chromosome is much longer and it has a lot more actual useful genetic information on it. And by the way, you know how chromosomes are numbered 1 through 23, and then you have two sets of 1 through 23. It's the 23rd chromosome that refers to the X and Y chromosomes. So the 23rd chromosome is what we're talking about here. Okay, and again, remember, they do form kind of tetrads. They do pair up during meiosis. Remember, tetrads are formed during meiosis. Well, tetrads will form here, but it's because of these pseudo-autosomal regions at the tips that allows them to form up to form a tetrad, not because they actually share a lot of regions of homologous regions. Okay, now there's even more interesting systems out there. For example, in birds, snakes, butterflies, some amphibian fishes, you'll have it's the male that has two of the same chromosome, and it's the female that has two different chromosomes. So, you know, again, in humans, XY, the two different chromosomes were XY, and that gives you males. But in these different creatures, the two different chromosomes would give you femaleness, right? So this is kind of an interesting fun fact to know. And again, from figure 4.5, we see the inheritance of sex in organisms with XY chromosomes results in equal numbers of male and female offspring. Why? Because, again, the male is the one with the X and Y chromosomes, and so the gametes can either be X or Y, and that is a 50-50 chance of an X or Y gamete. When the X gamete sperm fertilizes the female egg, which has to be X, you get a female. When the Y gamete sperm fertilizes the female egg, you will get a male. So it's a 50-50 chance. You always have a 50-50 chance to have a male or a female, but it's dictated by the sperm. The sperm dictates the sex of the offspring. Now, there are some interesting creatures out there as well, including some plants, some fungi, protozoans, fish, that don't have sex chromosomes at all, and they call these the Genic sex determination system, where there are no sex chromosomes. There's no X and Y or ZW or ZZ. It's only the only sex determining genes are found on autosomal. They're found on autosomal genes. And I don't know if I've defined this before, but obviously autosomal chromosomes are the non-sex chromosomes. So for humans, the autosomal chromosomes refer to chromosomes 1 through 22. The 23rd chromosome is called the sex chromosome. So in Genic sex determination system, there are no sex chromosomes, only autosomal chromosomes. And it's genes on the autosomal chromosomes that direct sex determination. Now, some creatures actually, it's very interesting, but temperature can direct sex determination. These are called environmental factors and their role in sex determination. So for example, turtles, warmer incubation temperatures produce more females during certain times of the year. But for alligators, the reverse is true. Isn't that interesting? Remember the plot line of Jurassic Park, that they should have all been females, but life found a way, as the doctor said, and that was due to temperature differences and the spontaneous change in sex. Okay, concept check number 2. How do chromosomal, genic, and environmental sex determination differ? Remember, we just covered all this. Chromosomal sex determination. There's a difference between the chromosome alleles of males and females. You have the XY, for example, in humans, and the XX in females. Genic, there are no sex chromosomes. Remember, there are only autosomal chromosomes, and sex determination occurs on those autosomal chromosomes. And then environmental, remember, environmental factors such as incubation, temperature, and time of year can affect sex determination. Now, again, in humans, you have the XX or XY system, right, for sex determination. The SRY gene, which I told you about, this is the important gene which gives maleness, right? This is the important gene on the Y chromosome that lends maleness to males. And it lives on the Y chromosome. Now, what's interesting is that humans can be born with different numbers of X and Y chromosomes. You could have, in Turner syndrome, you could have just an X chromosome, so you could be born with only an X chromosome, and that's it. No Y chromosome, no other X chromosome to complement. And this occurs in about 1 in 3,000 female births. Klinefelter syndrome, you have extra X chromosomes. You can have 1 extra, 2 extra, 3 extra. You could have an extra Y and an extra X, and this happens in 1 in 1,000 male births. Notice the presence of the Y chromosome directs maleness, and that's due to the presence of the SRY gene on the Y chromosome. The absence of a Y chromosome directs for femaleness, and that's why you would get the female births. Now, you can also have poly-X females, and that's when you have a bunch of X's. You could have 3, 4, 5 X's. Isn't that interesting? And that can happen in 1 in 1,000 female births. So here's an outline. Let's just go through this. Sex chromosomes, associated sexual phenotypes in humans. XX for female typically, which the characteristics are female traits. XY for male traits. Then you can have, like I said before, Klinefelter syndrome, XXY, XXYY, XXXY. This would give you the characteristics: male traits, tall, small testes, reduced facial, and pubic hair. Now, you can also have Turner syndrome, where, like I mentioned before, you could just be born with 1 X chromosome, and that's it. In this case, you would have female traits, you would be short, a low hairline, broad chest, and neck folds. Now, remember what else you could have? Poly-X females, 3 X's, 4 X's, 5 X's. These are known as poly-X females. Female traits, tall and thin. Interesting. Then you can have males with an extra Y. This is also very interesting. You can have XYY, male traits, tall. So very, very interesting stuff. And again, I was telling you about this SRY gene. You can see it here on the Y chromosome. It lives on the short arm of the Y chromosome. This gene is Y-linked because it is found only on the Y chromosome. And it directs maleness. Concept check number 3. In humans, what will be the phenotype of a person with XXXY sex chromosomes? What would be the phenotype? That's Klinefelter. Klinefelter, we touched on that. So these would be, will demonstrate Y characteristics, maleness due to the presence of that Y chromosome and the presence of that SRY gene. So now let's talk a little bit about these X-linked characteristics. What happens when you have genes that are on the X chromosome, and those genes may, develop a mutation or something? How does that affect males and females differently, and how would that differ from Mendel's patterns of inheritance? His typical monohybrid cross predictions. So let's look into this. All of these studies really started with this gentleman, Thomas Hunt Morgan, with his work in Drosophila melanogaster, where he had labs and he would grow up his fruit flies and he would look at the fruit flies and try to unravel the mysteries of heritability and genetics using these fruit flies. Remember, fruit flies are a very good model organism. What he actually found were he found a very rare mutation among the fruit flies. He found white-eyed fruit flies, which is very interesting because the wild type or normal phenotype is a red eye fruit fly. I mean, just think about it. When was the last time you saw a white-eyed fruit fly? It's very rare. So when he found these white-eyed fruit flies, he was very excited and he wanted to learn more about it. He wanted to know, well, is this a dominant trait? Is this a recessive trait? And he would set up these monohybrid crosses. And when he set up the monohybrid crosses, he was very shocked to see a pattern of ratios, inheritance pattern that differed from what Mendel predicted. What he predicted was a 3-to-1 phenotypic ratio, a 1-to-2-to-1 genotypic ratio, correct? But that's not what he saw. So let's take a look at what he saw. So what he eventually tried was separating these out by sexes, where you had the female with the wild type. You know, they called this X plus X plus, right? X plus meaning the X chromosome has the wild type allele and the other X chromosome has the wild type allele. So this is a true breeder wild type female, and then crossed it with a true breeder mutant male. So this is XW. W is for the white eye mutation. So, and obviously the Y chromosome doesn't have the mutation because the Y chromosome doesn't have an eye color gene. Remember, it's the X chromosome that has all these different genes. The Y chromosome has very few genes, and the main gene that's important for directing maleness is that SRY gene. So when you have meiosis and the gametes form, what kind of gametes can this female make? This female can only make gametes that are wild type. So the X chromosome has the wild type red eye allele. Now the male fly, remember male flies, males direct either Y chromosomes or X chromosomes into their gametes at a 50-50 chance. So their X chromosome has a W for the white eye, and the Y chromosome obviously doesn't have the mutation at all. So let's just follow this down first, then we'll come back and look at the reciprocal cross here. So let's take a look. So what happened? That wild type egg from the mom met either a wild type, a white mutant from sperm from the male, and that would give you this genotype here. That would give you red-eyed females, right? On the converse, the wild type female egg met a Y chromosome from the male sperm, and that would also give you red-eyed males. That would also give you the wild type males. So all of the offspring were wild type. Does that make sense? All of the offspring were wild type. But let's look at the reciprocal cross over here. When Thomas Hunt Morgan did this in reverse with the white-eyed female flies, this would mean they have XW genotypes, and cross that with the wild type male X plus Y. Remember, after meiosis, what kind of gametes could this true breeder make? Well, all the gametes have to be XW, and the male, you can have X plus or Y. So after fertilization, what could happen? If the XW egg meets the X plus sperm, you have XW X plus offspring, which are red eye females. However, if the XW egg met the Y sperm, you would have white-eyed males. Do you see how this would totally differ from Mendel's expected ratios for the P1 to F1 cross, the P to F1 cross? You see, so in this case, you would have half your fruit flies with red eyes, half your fruit flies with white eyes, and 100% of them with the red eyes are female, and 100% of them with the white eyes are male. You see that? So that should tell you right away that this is a sex-linked, they call these sex-linked or X-linked characteristics. That means that the gene or allele you are studying lives on the X chromosome, and typically that means that it cannot be complemented by a gene on the Y chromosome. So even if it's a recessive allele, it acts as a dominant allele. It can't be masked in males. Okay, very interesting stuff, right? Very interesting stuff. And then they went on, let's see, they went on to do the rest of the monohybrid cross. So let's start with this one on the left again. This female was X plus XW. That means she could make eggs with X plus or XW. This male was X plus and Y, so the sperm could be X plus or Y. So what did we see? You put the sperm information on top in this Punnett square, the egg information on the side on this Punnett square, and it looked like you had one red-eyed female that was X plus, one red-eyed female that was X plus Y, one red-eyed female that was X plus W, and then you had one wild-type male. So this looks to be a 3-to-1 phenotype ratio, which actually makes sense. You know, Mendelian, this would follow Mendelian genetics, right? 3-to-1 phenotype ratio. But when you do the converse, when you look at the converse cross, where this female was X plus XW, the eggs she could produce are either X plus or XW. The male could produce XW or Y. When you plot this out, you would see again half of the half of the flies have red eyes, half have white. So this is a 1-to-1 ratio of red to white eyes, and that totally deviates from Mendel's expected ratios, and again, once again shows that this is not a typical dominant recessive situation. Instead, this is likely an X-linked or sex-linked inheritance pattern. And again, why the fruit fly? The fruit fly is great because it is a great model organism. It's small size, it has a great reproduction time, it has a lot of offspring, etc., etc. This is the life cycle of a fly. You don't really need to know this in too much detail, but again, this just highlights why we use flies so commonly in population genetics, in all kinds of mutation research. I told you in stem cell research, chromosome variation, behavior, studying chromosomes, genetic control of pattern formation, you know, body segment formation, behavioral genetics, so many different aspects of life that we could study flies and directly relate those results to humans. And the whole reason we know about sex-linked or X-linked chromosomes is because of these flies. Very, very interesting stuff. And we can look at an example of X-linked inheritance in humans by looking at color blindness. I mean, most of you probably know that color blindness is more common in males than in females in humans. And here's why. It's an X-linked trait. So you have a normal color vision female, a human female. She would have X plus X plus. Remember, the plus means that it's a wild type allele on the X chromosome. Her gametes can be X plus or X plus. The colorblind male can have X with the C allele, which means colorblind, or Y. So the gametes, the sperms, could be XC or Y. And then after fertilization, you only have normal vision in the offspring. Normal vision. Why? Because if you have X plus XC, you have a wild type X chromosome in the females. And if you have X plus Y, you have normal wild type. So the wild type is dominant, and the wild type will compensate for the color blindness. So conclusion, both males and females have normal color vision. But in the reciprocal cross, this is where it gets interesting. In the reciprocal cross, so you have a colorblind female this time, a true breeder colorblind female. So she would have the two X chromosomes that have the C allele for colorblind. So her eggs can only possess the X chromosome with the colorblind allele. The normal vision male can make a wild type X chromosome sperm or a Y sperm. And when you do that Punnett square cross, you will see that all of the females have normal vision, but all of the males are colorblind. So do you see why out there, amongst populations of humans, you see now why there are so many more males that, have this colorblind trait than females? This because it's X-linked, and X-linked traits tend to affect males more than females because they don't have two X chromosomes with the ability of one of those X chromosomes masking or the effect of the other, right? They don't have a complement to help them out as females do. See here, in this female, the wild type complement was able to mask the colorblind. But in males, the colorblind X allele is the only one they have. There's nothing to complement on the Y chromosome. So concept check number 4, and then we'll take a quick little break time with Gizmo and Wicket. So let's get started. Concept check number 4, hemophilia, which is reduced blood clotting, is an X-linked recessive disease in humans. Kind of like color blindness then. A woman with hemophilia mates with a man who exhibits normal blood clotting. What is the probability that their child will have hemophilia? We just covered this, didn't we? Isn't it identical to this situation here? Instead of color-blind female, in this case, it would be hemophilia, right, in the female. So wouldn't it be 50-50? All of the females would be normal for blood clotting, and all of the males would be abnormal? Yep, exactly right. So again, on that note, let's take a quick little break time, and we'll be right back. Hey everyone, welcome back from break time with Gizmo and Wicket. Where we left off, we were talking about the X chromosome, and one thing you should realize is something very interesting with respect to the X chromosomes, and that is when you have more than one X chromosome inside of a cell, the excess X chromosomes inactivate themselves. So if you're a male, you only have one X chromosome, so the X chromosome is read by the cell. But if you're a female and you have two X chromosomes, well then one of those two X chromosomes will inactivate itself. It will form what is called as a Barr body. A Barr body is an inactivated X chromosome. And you can actually see it in the nucleus of female cells. And you don't see a Barr body in male cells. And why? Again, because they don't have to inactivate an X chromosome. So it doesn't matter how many X chromosomes you have, all but one becomes a Barr body. All but one gets inactivated. So let's look at this. If you have two X chromosomes, this is your female, no syndrome, and you have one Barr body. If you're a male, no syndrome, no Barr bodies. If you've got Turner syndrome, that you've got 0 Barr bodies. If you're Klinefelter with one extra X, you have one Barr body. Klinefelter 2 extra X's, one extra X and an extra Y, you have one Barr body. Klinefelter 2 extra X's, 2 Barr bodies, and so on and so forth. You see, so it doesn't matter how many X's you have, all but one gets inactivated and becomes a Barr body. This actually explains the patchy distribution of color on tortoiseshell cats This is due to random X inactivation. What does that mean? That means in the cat's body, certain cells have inactivated one X chromosome, which leads to one coat color, and other cells in the cats have activated the other X chromosome, which leads to a different coat color. So, and this usually happens earlier in development, this X inactivation, and that's why you see these patchy areas. It's not that one cell is different than the cell adjacent to it, it's that you've got this patchy orientation due to this X inactivation being random and happening a little bit earlier in development. Isn't that interesting? So here's concept check time. How many Barr bodies will a male with XXXYY, so Klinefelter with 2 extra X's and an extra Y chromosomes have in each of his cells? What are those Barr bodies? Okay, so you're going to have 2 extra Barr bodies. Each Barr body is an inactive X chromosome. Y chromosomes do not need to inactivate themselves because they don't have a lot of genetic information. There's the SRY gene mainly, and the Y chromosomes do not need to be inactivated. Now let's talk a little bit about the Y-linked characteristics. What's going on with the Y chromosome? These Y-linked characteristics are also known as Hollandric traits. They're only present in males from Y chromosome, which is inherited by the father. Remember, your Y chromosome comes from your father, and it's the father's sperm that dictates whether you have the XY male or XX female offspring. All male offspring will exhibit the trait. The Y chromosome has lost DNA over time, and the Y chromosome is mostly non-functional, so it accumulates mutations which serve as good genetic markers, or SNPs. The Y chromosome has lost a lot of DNA over time. A lot of the Y chromosome is non-coding information. And so it accumulates mutations quickly because it's not information that's genic. It's not information that's being used for anything. So you can actually look at those genetic markers, these SNPs, short nucleotide polymorphisms, and you can use that to trace your lineage back through your paternal past, if you will. It's pretty interesting stuff. And it's also important for sex determination. Remember that SRY gene is important for sex determination. It's the important gene that directs maleness in the Y chromosome for males. So now moving on from the sex-linked or X-linked inheritance, let's look at other types of inheritance that deviate or that appear to deviate from Mendel's mode of inheritance. Let's look at concepts like dominance, like complete dominance, incomplete dominance, and codominance. What Mendel was studying in his monohybrid and dihybrid crosses would be complete dominance. You have one trait that completely masks another. But you can also have incidents of incomplete dominance and codominance, and this can confuse the pattern of heredity that you can see. So let's explore this. So again, complete dominance is when the phenotype of the heterozygotes is the same as the phenotype of one of the homozygotes. So this is what Mendel saw. If you had purple flower plants and purple is the dominant color, well, it would always mask the white flower plant, and the F1 generation would always be purple flower. But what about incomplete dominance? What about that? Phenotype of the heterozygotes is an intermediate. It falls within the range between the phenotypes of the two homozygotes. Now, keep in mind, I'm going to show you this in more detail a little bit, but this is not the same thing as blending. Remember, blending hypothesis was disproven. Incomplete dominance is not blending, and you'll see why in a little bit. Let's look at codominance. Codominance, the phenotype of the heterozygote includes the phenotypes of both homozygotes. Again, this is not blending either. And a lot of times you can confuse incomplete dominance with codominance. So we're going to go through and talk a little bit more about the difference between the two. So here's complete dominance at the top left. Again, if you have a red flower plant and a white flower plant, well, if red is dominant to white, then the offspring are going to be 100% red, right? Now, oh, and not only will the offspring be 100% red, the only variability you will find in the offspring are either red flowers or white. You're not going to find pink or anything like that. Now, with incomplete dominance, you will find if the phenotype of the heterozygotes fall between the phenotypes of the two heterozygotes, dominance is incomplete. So in this case, you would find red, you would find white, and you would find everything in between. You would find different shades. Okay. So concept check time. How does incomplete dominance, complete dominance, incomplete dominance, and codominance differ, which is what we just touched on? But again, with complete dominance, the heterozygote expresses the same phenotype as one of the homozygotes. So you either have red flowers or white flowers. With incomplete dominance, the heterozygote has a phenotype that is intermediate between the two homozygotes. And with codominance, the heterozygote has a phenotype that simultaneously expresses the phenotypes of both homozygotes. Again, I know this is confusing, but one way to think about it is with codominance. Let's talk about incomplete dominance first. With incomplete dominance, you're going to have some kind of intermediate phenotype. So again, like pink flowers. Let's talk about codominance. Let's say there is a gene for an extra finger, and then there's a gene for a split finger. Well, it wouldn't be some mix of the two. It would be an extra and split finger. You would see both of those traits together. Does that make sense? They'd both express themselves, and they'd both present themselves, if that makes any sense. So with penetrance, here's another concept. Penetrance, this is the percentage of individuals that have a particular, that show a particular phenotype, that have a genotype. So just because you have a genotype, like you have an allele for something, doesn't mean you're going to express it. Even if it's a dominant allele, even if it's dominantly inherited, it doesn't necessarily mean you will, 100% of people will express that phenotype. This is penetrance. And then there's expressivity, which is the degree to which the character is expressed. So you can have traits that have variable penetrance. So some people don't show the trait even though they have the genotype for the trait, whereas some people show the trait because they have the allele for the trait. And then you can have variable expressivity as well, which means that, how defined is that trait? In some people, it might not be defined, very well defined at all. It might be a minor phenotype, and in others, it might be a major phenotype. So let's look at an example with human polydactyly. With human polydactyly, you have an extra finger, and the penetrance is about 90%. That means of the people with this human polydactyly gene, only about 90% show polydactyly. And of the ones that show it, of the 90% that show polydactyly, there's variable expressivity, which means you could have everything from a tiny little nub as the extra finger, or a tiny little skin tag, to an actual extra finger. So you see the difference between penetrance and expressivity. Penetrance is what percent of people with that allele express it, like show it, and expressivity is of those that show the phenotype, how pronounced is that phenotype? So concept check number 7, how does incomplete dominance differ from incomplete penetrance? So we talked about this. I'm not going to read through all of these, but let's look at the answer. In incomplete dominance, the heterozygote is intermediate between the homozygotes. So you've got, for example, the pink flower. In incomplete penetrance, some individuals do not express the expected phenotype at all, right? So that's a completely different outcome there. Here's another situation that you need to be aware of that can really affect the Mendelian ratios that you expect. And this is the case of lethal alleles. Believe it or not, yellow coat color in mice can be lethal and result in abortion, you know, basically a miscarriage in the mouse. So look at yellow fur color. If you're homozygous dominant for big Y, homozygous big Y, that's actually a lethal allele. It's considered a lethal allele. It results in a dead pup, right? And this is a miscarriage. This would not be a living birth, right? This would not be a viable birth. So what you would need to do for this Punnett square, you would take a non-true breeder, a heterozygous yellow mouse with a big Y, little y, and you would breed it with a heterozygous non-true breeder yellow mouse, big Y, little y, and after fertilization you would expect a quarter to be big Y, a half to be big Y, little y, and a quarter to be little y, little y. But if you were just focused on the offspring, if you were just focused on the live births and you didn't know this concept of lethal alleles, what would you say the outcome is? You would look at the pups and you would say, well, about 2/3 are yellow and 1/3 is white, so there's a 2-to-1 ratio of yellow to white, and that does not agree with Mendel's pattern of inheritance. But if you know the concept of the lethal allele, you would be able to factor in the quarter of the fetuses that didn't make it to birth, to fruition. So again, lethal alleles exist. There are combinations of alleles that you could inherit, and that would result in, you know, miscarriage. And you need to factor those in. You need to be aware that they at least exist, because that could mess up your studies. If all you're studying are the live births, you're going to miss the lethal effects of the lethal alleles. Okay, so keep in mind that there can be multiple alleles for a given locus within a population. So for example, you can have the ABO blood group, you can have not just blue or brown eyes, but you could have hazel eyes, green eyes. There could be more than just 2 alleles within a population. Also, keep in mind that frequently genes exhibit independent assortment themselves, but they don't necessarily act independently in their phenotypic expression. The reason for this is gene interaction. Gene interaction. This can confound heredity studies, because the effects of one gene can actually affect the phenotype of a different gene at a different locus. And there are 2 examples of this: gene interaction, which I'll explain in just a minute, and epistasis, which I'll also explain after that. So here's an example of gene interaction. And by the way, if Wiki's come to say hi and help me to do the presentation, he's just hanging out over here. Hi, Wiki. All right. Now, with his support, we will get through this example of gene interaction with the pepper Capsicum annuum. Now, here's an example of what I said, gene interaction. There's going to be a gene interaction that affects the color of this pepper. Notice that this is the P generation. These are the true breeders for red peppers and cream peppers. The reason this pepper is red is because it has 2 different pigment genes, two different dominant pigment genes, capital Y and capital C. Okay, and there's a gene interaction between those two pigments that gives the pepper the red color. And the cream color pepper, the reason it's cream color is because it's missing both of those genes, both of those pigment genes, or pigment producing genes, I should say. Now, when you cross the cream color pepper with the red pepper, you're going to see 100% red phenotype in the F1 generation. That's because these are hybrids, but they both have the capital Y and the capital C. They all have the capital Y, the capital C, so all of the offspring are going to be red because of the gene interaction between Y and C. But when you self-fertilize, the F1 generation and you look at the F2 generation, something interesting happens. It reveals the consequence of the gene interaction. Some of these are capital Y, capital C. In this case, you're going to have red peppers because of the gene interaction between Y and C. In some of these, in 1 in 16, you're going to have no Y and no C, right? No capital Y, no capital C, and these are going to be cream like the original parents. However, if you only have a dominant Y and only recessive C, you're going to see the phenotype of just allele Y. And the phenotype of allele Y by itself, without the gene interaction with C, is a peach color plant, a peach color pepper. Now, if you have just C and no Y, then you're going to see the phenotype of just allele C, which is an orange pepper. Does that make sense? So this shows you that allele Y gives you a peach color pepper, allele C gives you an orange color pepper, and the combination of the two gives you a new phenotype. This is a gene interaction to give you a novel phenotype, and that's exactly the definition of gene interaction. It produces a novel phenotype, a new phenotype, which is red. And the lack of either of those pigment producing enzymes is cream color. Okay, now let's take a look at a different example. Let's look at epistasis. This is where one gene masks, not works with, not interacts with, but one gene masks the effect of another gene, the hypostatic gene. So the epistatic gene is the one that does the masking. The hypostatic gene is the one that gets masked. And you can have recessive epistasis, or you could have dominant epistasis. So let's take a look at dominant epistasis. Here, you're dealing with the the W allele. This is the epistatic allele, and you have the Y allele. This is the hypostatic allele. Notice what Y does first. Y converts compound B, a green color compound, to compound C by making an enzyme, enzyme 2. But what if compound B wasn't there? Think about it. What if compound B was not there? If there's no compound B, then the enzyme that's produced by allele Y wouldn't be able to convert green to yellow. And that's exactly what this epistatic gene does. You see this capital W, this capital W epistatic gene, it produces enzyme 1, which converts compound A, a colorless compound, to compound B, a green color compound. So we could say that W is, capital W I should say, is epistatic to Y, because Y can only produce its phenotype, yellow squash, if W, capital W, is not around. Does that make sense? Because when capital W is not around, you're able to produce compound B, and then capital Y can convert compound B to compound C. So what is the conclusion here? Genotypes big W, big Y, and big W, little y do not produce enzyme 1. It makes sense because that's the epistatic gene, dominant epistatic. So you only need one. By the way, that's why it's dominant epistasis. You only need one copy of capital W. If this was recessive, you'd need 2 copies of that epistatic gene. Genotypes big W, big Y, and big W, little y do not produce enzyme 1. little little y produces enzyme 1, but not enzyme 2, so you're going to end up with green squash. And little w big Y produces both enzyme 1 and enzyme 2. That's the only way to get yellow squash. Does that make sense? Here's just a table outlining the different combinations we were talking about. Now let's move on to sex-influenced and sex-limited characteristics and define these terms here. Sex-influenced characteristics are those determined by autosomal genes, not the sex-linked genes, not the X-linked genes. These are determined by autosomal genes and are inherited according to Mendel's principles, but they get expressed differently in males and females. So usually it's the sex chromosomes have an effect on these genes, but these genes do not reside on those sex chromosomes. Then there are sex-limited characteristics. These are encoded by autosomal genes that are expressed in only one sex, and these have zero penetrance in any other sex. So they're only found in the one sex, even though, again, they are autosomal genes, which means they don't actually reside. Their locus is not on the sex genes, or the sex chromosomes, X or Y. And then there's cytoplasmic inheritance. This is where characteristics encoded by genes located in the cytoplasm occurs. So you could have genes in the cytoplasm, and that can give you a phenotype. Genes in the cytoplasm can also include genes of the mitochondria. So let's do a concept check. How do sex-influenced and sex-limited characters differ from sex-linked characteristics? Well, we just touched on that. Remember, sex-linked characteristics are found on the sex chromosomes, whereas the sex-influenced and sex-limited characteristics are found on the autosomes. So let's double check our logic here. Both sex-influenced and sex-limited characteristics are encoded by autosomal genes, whose expression is affected by the sex of the individual who possesses the gene. Sex-linked characteristics are encoded by genes on the actual sex chromosomes. So here's an example of cytoplasmically inherited characteristics with regard to the mitochondria. Remember, mitochondria have their own genes and their own chromosome, and that is considered part of your cytoplasmically inherited genes. Now, sometimes mutations can arise in these mitochondria, and over time, with each subsequent cell division, those mutated mitochondria can accumulate in particular cells. So you could end up with one cell with all mutated mitochondria and one cell with healthy mitochondria, and then you could end up passing on this mutated mitochondria to your offspring. So for example, what if this became an egg, right? What if this became an egg that was fertilized? In that case, this person would have mutated mitochondria, and there are actual genetic diseases, genetic disorders that you can inherit from such a case, from mutated mitochondrial genes, and this can have lifelong debilitating effects on the child. So again, cytoplasmically inherited traits present in males and females, usually inherited from one parent, typically the maternal parent, especially if you're talking about through the mitochondria. The mitochondria is only inherited by the mom's egg, through the mom's egg. So all of the mitochondria in your body, by the way, are from your mom, because the egg was the only gamete with mitochondria. The sperm didn't bring any mitochondria with it. So all of the mitochondria in your body are from your mom. And so if there was a mutation in those mitochondrial genes, then you're going to have problems, but it's because of the mutation that originated in your mom, not from the dad. Reciprocal crosses give different results, obviously, because usually you inherit that cytoplasmic DNA from one parent usually, and it's usually the mom. So a reciprocal cross would result in no effect. And exhibit extensive phenotypic variation even within a single family. Again, that makes sense because if you think about it, your sibling could have inherited all bad mitochondria, mutated mitochondrial genetics, and you may have inherited wild-type mitochondria. So you might be relatively healthy, whereas your sibling might be relatively sick because of the proportion of the mutated mitochondria that you versus your sibling inherited. Pretty interesting stuff. So let's get back to sex-influenced and sex-linked characteristics, going into some examples of each. So we got genetic maternal effect. This is where the phenotype of the offspring is determined by the genotype of the mother. We're going to give you an example of that in a minute. Then there's genomic imprinting, where differential expression of gene material depending on whether it is inherited by the male or the female parent. So you might inherit an allele, but if you inherit it from your mom, it might get silenced. Or if you inherit it from your dad, that gene might be silenced. And what does that mean? There's different ways of silencing genes. For example, you could methylate a gene, and so you add a methyl group to the gene, and that could cause the gene to silence itself. Okay, that's an example of genomic imprinting. And then epigenetics, where due to alterations to the DNA, again through methylation or to the histones, so that they shut down gene expression, that do not include changes to the base sequence, this can affect the way the DNA sequences are expressed. When people talk epigenetics, they're usually talking about turning on or off genes, but not by changing the gene, but by changing the expression of the gene, usually by methylating the gene or by altering the histones. If you modify the histones, remember the histones in the chromatin, you can modify the little histone tails to either open up the gene so it can be read, or close up the gene so it can't be read, to silence the genes. So let's go through these again and define each one. We've touched on these before, but sex-linked characteristics, these are genes located on the X chromosome, the sex chromosome. Sex-influenced characteristic, these are genes on autosomes. Remember, autosome means any chromosome that's not the sex chromosome, that are more readily expressed in one sex. Sex-limited characteristics, very similar to sex-influenced. However, in this case, it's an autosomal gene whose expression is limited to one sex, not just more readily expressed in one sex. It's only expressed in one sex. Then there's genetic maternal effect. Remember, this is where nuclear genotype of the mom determines the phenotype of the offspring. Cytoplasmic inheritance. This is where cytoplasmic genes, which are usually inherited entirely from only one parent. For example, remember, the mitochondrial genes are an example of cytoplasmic genes. Any genes, any genomic material that's outside of the nucleus is called cytoplasmic genes. So if it's not part of your genome proper, if it's not one of your 46 chromosomes, it's a cytoplasmic gene. And all of us have DNA, a whole chromosome, a circularized chromosome of DNA inside of our mitochondria, and that's not considered part of our genome. That's cytoplasmic DNA, and we inherited that cytoplasmic DNA from our moms. And remember, if you have mutations in that mitochondrial DNA, you can actually have severe heritable issues, and they have to do with the misfunction of the mitochondria. Okay, and genomic imprinting. This is where genes whose expression is affected by the sex of transmitting parent. So again, like I said, you could inherit an allele from your mom, but it might be silenced due to a methylation or something, just by way of being inherited by that parent. So here is an example, an elegant example of maternal effect in the snail. you can have left-handed sinistral snails shells and dextral right-handed coil on the snail shell. In this case, you've got males with right-handed coils, right-handed is dextral, and female with left-handed sinistral shell. Now remember what maternal effect means. It means that the genotype of the mom determines the phenotype of the offspring. Okay, so follow that. The genotype of the mom dictates the phenotype of the offspring. Just remember that, and it'll make much more sense as we go through here. So here's what we're seeing. In dextral, which is right-handed coiled father, you have S plus S plus. Dextral, a right-handed coil results from an autosomal allele S plus that is dominant. Remember, S plus is dominant. The female, in this case, is sinistral, which means left-handed coil, which codes for sinistral, S, which is the recessive for left-handed coil. Now, when these parents mate, their gametes, S plus and S, fertilize, fuse to form offspring, F1 offspring. And get this, all of the F1 offspring are sinistral, left-handed coil, even though all of them are S plus and S. What, if you were just looking at the genotype, what should the phenotype be? If you were just looking off of this genotype, it's S plus S. Remember, S plus is dominant for dextral. This offspring is S plus S. So shouldn't this offspring be dextral? It should. It should be dextral because dextral is the dominant allele. However, it's sinistral. And the reason is, like I just said, maternal effect. The genotype of the mother determines the phenotype of the offspring. So let's see what it says here. All the F1 are heterozygous S plus S because the genotype of the mother determines the phenotype of the offspring. All of the F1 have a sinistral shell. So let's look at this in closer detail here. Again, the dextral dominant father cross with the sinistral recessive mother to give you heterozygotes that should be dextral if you're looking at it genetically, but are actually sinistral if you're looking at maternal effect, if you're factoring in maternal effect. Now, what's the next step? Self-fertilization, which means that this snail will have to mate with its own genotype, so it can make gametes with S plus or S. And here's what happens. You have offspring, but guess what? All of the offspring are dextral. All of the offspring are dextral. Now look at this. A quarter of the offspring are S plus S plus. That makes sense that they're dextral, right? Because S plus is dominant and S plus is codes for dextral. The heterozygotes are also dextral because S plus and S, well, S plus is dominant and it codes for dextral. So why then, think about this, why is this shell dextral? Why is this shell dextral? Little s should code for sinistral, but it's showing dextral. Well, remember what maternal effect means? The genotype of the mom, not the phenotype of the mom. The genotype of the mom dictates the phenotype of the offspring. So even though the offspring, 1/4 of the offspring should be sinistral, it shows dextral because of maternal effect. Very interesting stuff. So again, conclusion, because the mother of the F2 progeny has genotype S plus S, all of the F2 snails are dextral. Very interesting stuff. Now moving on to genomic imprinting. Again, remember, this is when you inherit an allele from one parent, but that allele might be turned off, switched off, due to methylation or some other kind of imprinting on the genome. So in this case, it's IGF-2. It's a growth factor for mice. The paternal allele has IGF-2. The maternal allele has IGF-2. The paternal allele is active, and its protein product stimulates fetal growth. The maternal allele is silent. It's switched off. It's imprinted off. The absence of its protein product does not further stimulate fetal growth. And then the size of the fetus is determined by the combined effects of both alleles. Isn't that interesting? So here's an example of genomic imprinting, where you're inheriting a gene, but it is switched off depending on which parent you inherited from. Humans also have an IGF-2 allele, and it is also imprinted, and it can affect fetal growth. So very interesting. Another example of how animals can lend to human studies. Now this one's really a favorite of mine. It's very interesting, and that's other types of phenotypes that are perplexing until further researched. And here's an example: temperature sensitive alleles. This is an allele whose product is functional only at certain temperatures. Notice this bunny. Isn't it black and white? But notice, where is it black? Where is this bunny? Where does it have black fur? On its ears, on its nose, on its little feet, and it's on its little tail, right? Aren't those the coolest parts of the bunny, temperature-wise? The ears are cool, the nose is cool, the hands, the paws, the tail. Here's the thing. The enzyme that produces this pigment in the fur is only functional at below 25 degrees C. So around room temperature. Room temperature is 24 degrees C. Does that make sense? So room temperature is around 24 degrees C. Your body's temperature, well, for humans anyways, around 37 degrees C. So, you know, if we had a similar enzyme, yeah, the tips of our ears might turn black too, you know, or the hairs might turn black. And that's because of the cooler tips of your fingers, tips of your nose, tips of your ears. Very interesting stuff. At any warmer than that, and the enzyme unfolds, right? So these are temperature sensitive mutations, temperature sensitive genes. So when the body temperature warms up a bit, the protein that's the result of this gene, the protein might unfold a little, and that protein unfolds, it may not function, and so it can't produce the pigment in the rest of the bunny. Isn't that interesting? All right, I believe this is the last concept of the chapter: polygenic characteristics versus pleiotropy. Polygenic characteristics, simply put, are characteristics encoded by genes at many loci. So for example, people often ask me, you know, Dr. D, what's the gene for height, for example? You know, like my brother's 6'5" and I'm only 5'5". You know, what happened there? What gene did I not get? Well, here's the thing. Height, for example, is an example of a polygenic characteristics. There are characteristics encoded by genes at many loci. There are many different genes that dictate your height. Does that make sense? Some of them dictate how long your bones will get. Some of them dictate how long, you know, how much growth there is during development. Right? There's growth factors involved. There's bone stimulating factors, bone morphogenetic proteins. There's all kinds of different genes that need to work in concert for you to be tall or short. It's not one gene. It's not like, you know, sickle cell that has one gene and you have sickle cell. There are many different genes involved here. And then pleiotropy, where one gene can affect multiple characteristics. You could have one gene play a role, let's say, in heart formation, and then it can play a role later on, maybe in blood vessel formation too. So you have one gene and it might have multiple different roles in development and then in the function of the body. So again, what is the difference between polygeny and pleiotropy? Polygeny refers to influence of multiple genes on one characteristics. Pleiotropy is the effect of a single gene on multiple characteristics. All right, and that leads us to the end of this chapter. I hope it made sense. This is a little bit of a confusing chapter, but nothing you can't get through. I hope you guys learned a lot, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.